junior high-ish, because I think that's probably like when it started to really, my eyes were like,
for physics.
just like super weak.
Yes, they were graded.
Yeah, we had graded
Like,
one
wow,
too. That sucks.
my mom like walked in on me when I was sleeping.
The 5-credit one before it changed to the bio one.
Okay, I had the 5 credit.
Oh, I had the 5-credit one. Yep.
Before it was the 5. Okay.
And then it was like the one for like CBS and
Everyone, please sign your tablet or mobile device or what have you on into this session ID if you haven't already done that. I get really distracted by stuff out in the halls, so I try to keep the doors closed, but let me know if anybody's out here trying to get in. Okay, how is everybody today? Can you hear me all right? Still happy with your life choices after a few days of... All right, good, I'm glad. Okay, so we left off on Monday with a discussion of this problem, right? Did you have a chance to reflect on this at all? Yeah. Okay, so this was how the question appeared, right? And I, just in the interest of time, let's kind of start to dissect it right away. Okay, so should be pretty simple here, right? So at this point in your medical school career, think of pathologies in the context of disruptions of normal physiology. Okay, the most important thing for you to learn over the next 2 years is what normally happens in a healthy individual. Okay, and if you have a good understanding of those processes, then you overlay the pathology on top of that. Okay, so that's just my unasked for advice there. Okay, so anemia means paucity of red blood cells, so probably red blood cell homeostasis is a good answer. Okay, so let's look at that then. Red blood cell, the number of red blood cells that are circulating in your blood here, this is going to be reflective of the rate of synthesis of the erythroid precursors and their maturation into red blood cells in the bone marrow. All right, so that's going to be one end of it. Then another end of it is the destruction or the removal of those red blood cells from the circulation in the spleen. Okay. And then intermediate to that, we could also have just loss of blood in circulation from either physiological bleeding or wound or traumatic bleeding. So those three processes are going to be the aggregate of those three processes are going to determine whether the amount of red blood cells in your circulation is normal or abnormally high or abnormally low. Okay? So let's look at this a little bit more detail. So the patient had a macrocytic anemia, right? So macrocytic anemia is referring to, well, we know what the anemia means. Macrocytic means large cells. Okay, so there are few red blood cells, but the ones that are there are abnormally large. So that gives us a clue as to the origin of the underlying cause of the anemia. So the size control of the red blood cell is determined during the process of division and maturation of the erythroid precursor cells. Okay, so in the bone marrow, you have a erythropoietic precursor, somewhat pluripotent stem cell that divides into a erythroid precursor. Then that divides into daughter cells that mature into red blood cells. Okay, they poop out their nucleus and their mitochondria, and then they become red blood cells. So that size control of the cells happens in development, and the size is determined in normal circumstances, when there is no limiting substrate for, or limiting substrate that's required for division, the control of the size of the red blood cell is tightly coordinated with the replication of the genome. Okay, so the cell cycle and accumulation of mass in normal development of erythroid precursor cells is tightly coupled. Okay, so they get to be so big, and as they're getting to be so big, they're replicating their genome into four copies of each chromosome, and then they physically divide into two daughter cells that are appropriate size. So if you have cells that are too large, that suggests that there's something here that's making them accumulate mass more than they should. And the cause there is a problem with replication of DNA. So the replication of the genomic DNA that would precede cell division is delayed in this case. So the cells are accumulating more mass before they ultimately divide than they should. Does that sound right? Okay, so I'm going to step on some of Dr. Cormier's material. What are the requirements of DNA synthesis? Right, we need a template. That's there, right? That's within the cells. We need DNA polymerase. That's probably there, right? Nucleotides, that's something that the cell is going to need to synthesize during S phase and make them available to the polymerase, right? And they need how many of them? Billion? Few billion? Right, so they need them in large amounts. Okay, so let's consider where those nucleotides come from, and let's look at least at where one of those nucleotides comes from, the deoxythymidine monophosphate. So the thymidine nucleotide, or deoxythymidine nucleotide, this tends to be the rate-limiting nucleotide for DNA synthesis. And the synthesis of the thymidine nucleotide is dependent on the substrates of the deoxyuridine monophosphate. So you have a uridine monophosphate. The thymidine synthase enzyme, or the as shown here, I think is thymidylate synthase enzyme. It's same name for the same enzyme, different name for the same enzyme, excuse me. That needs a methyl donor. So the conversion of the uridine base to the thymidine base is a methyl transfer reaction. Okay, so the donor for that methyl group is derived from a vitamin that comes ultimately from folic acid. Okay, so the folic acid ultimately becomes tetrahydrofolate. That tetrahydrofolate takes a single carbon from the amino acid serine and then donates that carbon to the uridine monophosphate to make the thymidine monophosphate. Then this thymidine monophosphate, then this can be phosphorylated to the diphosphate and the triphosphate form, and then it's a substrate for the DNA polymerase, and that can then sustain replication of the genomic DNA in erythroid precursor cells and in any other cell that is undergoing cell division. Okay, whether it be a cell of blood origin or a cancer cell and pathological consequences or what have you. Okay, so significance here. This is a biochemical pathway that is supporting a normal physiological process of erythropoiesis. Okay, so we've got the folic acid. This is converted to the dihydrofolic acid and then tetrahydrofolic acid within the body. But that tetrahydrofolate, that's something that we can't make on our own. We need to eat it. Okay, bacteria or need to make it, and then we need to eat those, and then that's how we get it. Okay, so this is an example of a vitamin precursor that's required for normal physiology. and where we get the folic acid, mainly in leafy green vegetables, but also in yeast and a variety of other sources. Okay, so this is the normal physiology, all right? And in our case, we have a macrocytic anemia that results from a lack of folic acid. Okay, so how do we get the folic acid? If you recall, what was significant about our patient's history? Yeah, well, I'd say two things. First of all, she's an adult, right? Now, there are inborn errors of folic acid metabolism, like if you don't have the right transporters for folic acid in your digestive tract, you could get a macrocytic anemia. However, that's going to manifest itself in infancy, right? So that's kind of, one kind of thing to consider. But the alcohol abuse, yep, that was definitely significant, and you don't want to assume a whole lot about your patients, but you've got even in a long stem multiple choice question, you've only got a certain amount of space, so kind of assume that everything there is pretty significant. Okay, so in the case of alcohol abuse, especially if it's chronic and sustained over a long time, that can impact folic acid metabolism and frankly, the processing of many of the macronutrients and the vitamin micronutrients. And this has to do with maldistribution of fat, so any sort of a fat soluble vitamin could have a potential to be problematic. Corruption of the intestinal lining, so you get a lot of issues with absorption and distribution of nutrients. There's also oxidative stress that's associated with metabolism of large quantities of alcohol, and that can deplete the NADPH that's necessary to sustain the cycle of recycling the vitamin-derived folic acid. And then if you kind of extend this further into more sustained and severe alcoholism, there tends to be a monotonous diet. You're getting most of your calories from alcohol and you're not eating much of anything else. So all those things kind of compound to exacerbate that nutrient deficiency. Okay, now there will be a lecture on folic acid metabolism and vitamin B12 metabolism later on in the course. So, learn this now if you want to, but I'm kind of giving you this material now more as an approach to solving biochemical problems. Okay, so I may, give you a pathway and kind of have you deduce some stuff, but as far as the details of folic acid metabolism, don't worry about that so much for this first exam. Okay, fair enough. And I'm going to say a little bit more about all this stuff in a little bit. Okay, so let's go back to this question. So this is exactly how it was given to you yesterday. And I'm giving it to you again just to remind us, and then also I think a handful of respondents yesterday selected cobalamin. You remember that? Did anybody who selected that want to describe why they selected that? No, just guessing. Okay, all right, fair enough. Okay, so I think now we're going to have 100% folic acid on this, right? Okay, so the next best answer would be vitamin B12 or cobalamin. Okay, vitamin B12 deficiency is also associated with a macrocytic anemia. So what would be the key phrase in here that would point you away from vitamin B12? Anybody know? Yeah.
Normal neurological exam.
Yep, that's That's exactly it. Yep, normal neurological exam. So with the vitamin B12 deficiency, you get the macrocytic anemia, but you also get additional neurological symptoms that are associated with lack of neurotransmitter synthesis, for example. So vitamin B12 deficiency, this causes a functional folic acid deficiency. So the metabolism of those two are tightly intertwined. Okay, so when you have a lack of vitamin B12, that's trapping all of the tetrahydrofolic acid pool in a different oxidation state of the carbon, and then it's not available for thymidine synthase. Okay. Okay, so anyways, that's just stuff that is going to be second nature to you by the end of the 2 years here in Duluth. Okay. All right, so I'm going to just go back to this question. This is essentially a one-step question. Have you heard people talk about one-step versus two-step questions before? Yes, or like in orientation at all, did they talk about that? Or?
A little.
A little, okay. So this is a one-step question, and this is a two-step question. Okay, so everything in the top, I think, is identical. It's just the what it's asking is different. And I think it's, not a super difficult two-step question, but you have to kind of recognize the clinical presentation as a folic acid deficiency, and then understand the pathology of it. Okay, good. Should we try another one? Okay, good. So this is getting a little harder. I mean, this is frankly something that you're going to cover in your last course here in Duluth in endocrinological and reproductive medicine. But just giving you some examples of where we can go here and how we can get, so best answer would be the neural tube defects in offspring. Okay, so that's associated with folic acid deficiency during pregnancy. And this is one that we would get to, I think, in the immunology, hematology, oncology course later in the spring. So if you have a lack of nucleotides that are available in your bone marrow for the erythroid precursors, you probably have a lack of nucleotides for all of the other cells in the blood as well. Right? So you can get a, what do they call that? Pancytopenia. Right? So not only a loss of red blood cells, but also a loss of platelets, for example, that would result in bleeding disorders. And shown here, same question, basically just one different answer, opportunistic infections. This could be due to a lack of lymphocytes because the lymphocyte precursors, T cells and B cells, are not there. Okay, so you can get a lot of mileage out of kind of the same concept, right? So any kind of pathology has a lot of normal physiology behind it, and a lot of different disciplines, not just biochemistry, but clinical practice and physiology, microbiology, etc. Okay, so think broadly about these issues. And I just want to, I'll give you this one just as an example. So this is a how you would present a vitamin B12 deficiency. Okay, kind of some of the same symptoms like weakness, macrocytic anemia. However, you have episodes of confusion, and then you also have this dietary history that if you know where the sources of vitamin B12 are in your diet, then that would kind of give you a clue. So vitamin B12 comes from foods of animal origin. And then this is a hard question. So the best answer is homocysteine. And yeah, hyperhomocystinemia, this is associated with a vitamin B12 deficiency and has some other problems as well. So anyways, don't commit all of this to memory, but I just want to give you a precursor of what you're about to face. Okay. Okay, so let's look at, just spend a few more moments looking at an approach to studying biochemical pathways, and I'll use this one that we talked about, the thymidine nucleotide synthesis pathway, as an example. So one approach is to memorize every substrate and every enzyme, and if you want to do that, or if you've already done that, don't want to discourage you from doing it, but it's not necessary to do it, and if you do that, you may be missing some of the context and the significance. Okay, so I'm going to give you kind of my systematic approach for success in studying biochemical pathways. Okay, so first thing is to look at the broad function of the pathway. Okay, use that teleological argument, imagine you are a cell in the body, why are you doing this? Okay, what does this support? Now, in the case of the thymidine nucleotide synthesis pathway, the big function is this makes nucleotides, and rapidly dividing cells need nucleotides for DNA synthesis. Okay, so just have like a one-sentence description that you have solidly at your disposal for each pathway. Okay, then the key substrates, in this case, the uridine monophosphate, the serine, this is the amino acid that is the ultimate carbon donor for that methylation reaction, and then NADPH. So the significance here will be appreciated shortly. This is a key source of reducing energy within the cells. Then we'll look at the key products. The key products here are, of course, the thymidine, deoxythymidine monophosphate, which is then available for phosphorylation to the triphosphate form and available for DNA synthesis. We also have the oxidized form of the redox cofactor NADP+. So these substrates and products, these will always go together. So that'll be pretty easy to remember. And then glycine, you take one carbon away from serine and you get glycine. Okay. Then we'll look at study the regulatory mechanisms. And for many pathways, there will be one single enzymatic step that is regulated and sometimes regulated in multiple fashions. Just to fill this out, the regulation of I should back up here. Okay, so the regulation of every pathway is going to be determined by the availability of the substrates and how much product there is, okay, just by energetics. Okay, so kind of keep that in mind. Irrespective of that, though, there can be regulatory steps that can determine whether a pathway is on or off independently, largely of substrate and product concentration. In the case of the thymidine nucleotide synthesis pathway, it's regulated by transcriptional regulation of the dihydrofolate reductase enzyme and the thymidine synthase enzyme. So those enzymes are only expressed during S phase when the cell is about to divide. Okay, drug targets. Red blood cell precursors aren't the only cells that want nucleotides. Some pathological cells want nucleotides as well. So cancer cells are rapidly dividing cells. And one therapeutic approach to stopping cancer is to deprive the cancer cells of nucleotides. Okay, so methotrexate, this is an inhibitor of dihydrofolate reductase, and then 5-fluorouracil, this is a analog of the uridine monophosphate that inhibits the thymidine synthase enzyme. So both of these are used in the context of cancer treatment. Methotrexate has some other applications as well. Okay, and then causes and symptoms of pathway dysfunction. We know if you don't have folic acid around, or if you have a problem with its metabolism, then you will have a paucity of nucleotides in erythroid precursor cells that will result in an anemia that will be macrocytic in nature. Okay, so I guess, if you add all that up, it's a lot. Whether it's easier or harder than memorizing every enzyme and product, I think it's easier. And also, it's going to become easier the more of that Stanford pathway that we fill out. Okay, so the biochemical pathways are going to start out as kind of, being isolated pathways that don't have any relationship to anything, but the more of them we learn, we'll see how they're related to one another, and I think it gets a little bit easier as you go along. Okay, so be patient and bear with me. Okay. Okay, final thing that I just want to note is this point, that if you have a problem with a biochemical pathway, the symptoms are generally going to result from either a lack of the product of that pathway or from an accumulation of an intermediate that is toxic. Okay, so for example, if we go back to our thymidine synthesis pathway, the pathology here is caused by a lack of the product of the pathway. Okay, we don't get any, pathologic effects from the accumulation of this, but in some cases, some pathways like heme synthesis, for example, if you have a disruption in that, you get accumulation of intermediates that are toxic. Okay, so just kind of keep that big point in mind. Okay, fair enough? Any questions? Okay, if you think of any questions, you have a moment between now and when I switch PowerPoints. Oops. Okay, we're back. So the topic for the next session is amino acid biochemistry and protein structure, and how protein structure is related to function. Okay, so I'm going to have to switch hats here and remember what we're talking about. Okay, so how we interpret these objectives and the level of detail that I'm going to expect, I'm going to maybe elaborate on that in a couple of places as we go on. Okay. Okay, first, should we do a little clicker quiz here? Okay, best answer is carbohydrates, protein, and fat. Very good. Okay, so triacylglycerol, that's our main dietary fat, and if you look at the carbon there, the carbon is very reduced. Not a lot of oxygen for every carbon that comes in. So that has by far the most caloric density of any of our foods. It also is the biggest source of stored energy within our bodies. Okay, very good. So the 2-carbon carboxylic acid, that's the acetate or acetyl portion. And then the vitamin-derived handle, that would be the coenzyme A. Okay, very good. And the significance there is that that's a key intermediate in many fuel oxidation pathways and in many biosynthesis pathways. Okay, so a few little easy one-step questions to get us started here. All right, so important classes of molecules in biology. There are 3 important macromolecules, if you will, or big polymers: carbohydrates, proteins, and nucleic acids. And then lipids, those join together in big aggregates that aren't polymers technically, but they are, big aggregations of fat molecules. So the discussion for today is centered around proteins because proteins are key for structure in cells, and they also do all of the biochemical work, or the majority of the biochemical work within cells. So the topic that we'll focus on here today is on proteins, and proteins are made up of individual amino acids. So we'll start with an introduction to amino acid biochemistry. Amino acids, the basic structure of an amino acid is just as the name suggests. So there's an amino group, the nitrogen and the protons around it, and then there's an acid group. In this case, the acid is a carboxylic acid. So those 2 groups are linked by a carbon, and that carbon we refer to as the alpha carbon. So the amino acids that are used to make proteins are all alpha amino acids. There are also some, amino acids where the nitrogen is hanging off of the R group down here, but those have other uses, not in making proteins. Okay, so amino acids, there are some intrinsic properties that are shared by all amino acids that are imparted by the chemistry of the amine group and the carboxylic acid group linked by that central carbon. Okay, so all amino acids share some properties that are derived from this part of the molecule. As shown here, the R group, the part of the molecule that's attached to that central carbon, that provides the variability or the diversity of the biochemistry in different amino acids. It allows us to group amino acids into different classes, and that also provides essentially all of the structure and function of any enzyme is due to the diversity of the R groups and the appropriate chemistry, the appropriate R group chemistry being in the appropriate place relative to everything else within the protein. Okay. So the functions of amino acids, of course, I'm going to focus on their role in, as in the the process of translation and post-translational modification, their role in the structure of proteins. However, there are a lot of other functions. The oxidation of protein can be used as a metabolic fuel, just like carbohydrate or fat. The amino acids are also key for nitrogen balance within the cells and taking nitrogen from one tissue to the next. Glutamic acid is an important neurotransmitter, and then amino acids are involved in the synthesis of other amino acids, and then just generally biosynthesis. So amino acids are, as in the example of the thymidine synthesis pathway, the serine amino acid is a carbon donor for thymidine synthesis, just as an example. So there's a zillion other examples. Okay, but I'm going to focus on their role in being the building blocks of proteins for this lecture. Okay, so let's see. There are 20 or 21 different amino acids, depending on what you count as an amino acid, that make up the proteins in the body. And I'll talk about the properties of the different amino acids grouped by their side chain chemistry in a moment, but first I'll talk about some of the shared properties of all amino acids that are derived from those side chain, by the carboxylic acid in the amine group. Okay, and I'll probably go through this pretty quickly because I think you've had it, I would hope, in other contexts. Okay, so you'll notice here there's a plus on the amine group and there's a minus on the carboxylic acid group. Okay, so this is showing the ionization state of the nitrogen and the carbon, the carboxylic acid, at neutral pH or near neutral pH. So if you were to take a pure amino acid in water and then add a ton of acid into that water so that it was at a very low pH, you would have the carboxylic acid would be protonated or deionized, I guess. Okay, so this would be in the protonated or uncharged form. The amine group, this likewise would be protonated as well, because in acid conditions, the definition of acid is, or the definition of low pH is high hydrogen ion concentration, right? So here, everything is saturated with protons. If you were to titrate in base and increase the pH to approximately neutral pH, here the first thing that's going to ionize is the carboxylic acid. This will then be in the minus form. The amine group, this is still going to be in the positive form. If you increase the pH well beyond physiological pH, then there you will then deionize the amine group and you'd have a net negative charge. Okay, so if you just consider the carboxylic acid and the amine group, at neutral pH, there's a neutral charge or a net neutral charge, 1 minus and 1 plus on an amino acid. Okay? And just, so this is a titration curve for a acid with a single ionizable group, acetic acid. Right, so just showing that titration curve, you have one pK value because you only have one ionizable group. In the case of an amino acid group, an amino acid, this is showing the relationship between the addition of hydroxyl ions, or the increase in pH, and the actual pH of the solution. So this pK value, this is at the low pH, this is reflecting the ionization or the balance between the ionized and the unionized form of the carboxylic acid. So in the case of glycine, the simplest amino acid, at the pH of 2.3, you're going to have 50% of the amino acids, carboxylic acids, will be ionized and 50% will be unionized. Okay, so that's the definition of the pK. If that ever comes up, I'm not sure if it will, but okay, and then, okay, I think the important thing here is to relate this graph with this graph. And is that clear, or does that need some elaboration? Okay, so this part of the curve, or this part of the reaction is shown here, and then this part of the titration curve is the ionization of the amine group. Clear? Okay. All right, so don't want to make too big of a deal of that. I think the importance is for the subset of amino acids that have an R group that has an ionizable group, or a group that is ionizable at near physiological pH. In that case, those amino acids have a third pK that's intermediate between the pK of the carboxylic acid and the amine group. And for these amino acids, for example, glutamate or aspartate would be among them. These are important because they can be in that different charge state stably at near neutral pH, and they frequently will participate in catalytic reactions because of that stability in the different ionization states. Okay, and I'll elaborate on that in a little bit. Okay, so what so measurement of these pKs, this is a very, it's not real, it is real, but it's pretty contrived relative to the behavior of amino acids within our body. Okay, this is looking at isolated amino acids in pure water and only modifying the pH. Okay, so that doesn't really happen within our body, and we wouldn't want it to probably. Okay, so within our body, amino acids exist or can exist in the context of a protein, so that the chemistry of any amino acid side chain and its backbone is going to be determined not just by its own properties, but also by the chemistry of adjacent amino acids. Okay, that whole big structure can contribute to any individual amino acid's chemistry. Did Cormier talk about translation yet, or is that coming up? That's coming up. Okay, so the process of translation on ribosomes will join one amino acid to another, so join the amine group of an amino acid to the carboxylic acid of another amino acid in a dehydration reaction, and that then makes a peptide bond, which is just the type of an amide bond that happens between amino acids. So the sequential linking of amino acids in this linear polymer, this makes proteins. And of course the precise sequence of the amino acids, that's determined by the codon sequence of the mRNA, and then ultimately by the DNA that is encoding the gene. So within the body, this is an enzyme catalyzed reaction. So both the formation of peptide bonds by the ribosomes and the breaking of peptide bonds by different protease enzymes, these are catalyzed by enzymes. So it's not a spontaneous thing that happens. Okay, if you'll notice the individual subunits of a protein, the amino acids, these have a polarity, right? They have a negative end and a positive end. Okay, so just as an example, the individual Lego blocks, each one has a nubby end and a recessed end, and when you put them together into a big polymer, the big polymer also has a nubbly end and a recessed end, right? So, okay, same thing with a protein. The individual amino acids have polarity. They're linked end to end to end, always in the same way. So the bigger structure also has a polarity. So when we talk about proteins, we'll talk about the amino terminus and the carboxylic acid terminus, right? Or domains of a protein that are near the amino end or near the carboxylic acid end, for example. And this, that's where that comes from. Okay. All right. Any questions? Okay. Okay, so that is a little discussion of kind of the intrinsic properties shared by all amino acids. And as we're talking about protein folding and development of secondary structure, I'll kind of refer back to that. But for the next little bit, I'd like to talk about different categories of amino acids. Amino acids can be categorized in a lot of different ways. For example, like whether they're required in our diet or whether we can synthesize them from simple precursors. You could categorize amino acids by hydropathy index, or I don't know. There are many ways you could categorize amino acids. I'm going to use the categorization system from your textbook. And I think it's pretty good. So it's categorizing the amino acids based on the chemistry of the amino acid side chains. And I'm going to go through each one of these each one of the categories, and I'll say a little bit about the broad properties of that category, and then some, little trivia properties of individual amino acids within that category. So if you refer back to the objectives, the way that I'm interpreting the first objective is that I don't want you to memorize the structure of amino acids, certainly, but I think if you're given an amino acid, you should be able to know what class it's in and then know some general things about that class. Okay, so that's kind of the first thing. There are also some post-translational modifications that we'll discuss, and you should also know which post-translational modifications happen on which amino acids. Okay, so serine, for example, you should know that that's a polar amino acid but not a charged amino acid. So what about that? Okay, polar amino acids. If an enzyme or if a protein is in a water environment, which most are in the cell, then a polar amino acid is probably going to be on the surface of the protein interacting with the water, right? Okay, so that you should know like that level of detail. Then the serine also is a site of a post-translational modification. It can be phosphorylated. Okay, so that's kind of the level of detail, and it's not. Don't want to minimize it. It's going to require some study if you haven't done this already, but it's not so daunting if you just consider that. Okay, let's see. Why don't we start off with the aliphatic amino acids, and then we'll take a little break. Okay, so aliphatic amino acids. If you're looking at the R group or we consider this the R group or the side chain. I guess those are two, ways to consider that variable portion in amino acids. If you look at the carbon on the side chains of the aliphatic amino acids, it's in a very reduced form. That is, there's not a lot of oxygen, and there's a lot of saturation with protons. Right, so it's almost like they're fat. Now that aliphatic nature, that's going to make the amino acids not want to be near water. Okay, so they tend to have the high hydropathy index. So that's like the general properties of the class. They tend to be in the interior of proteins. in the folded structure of proteins because they don't like the water, and that exclusion of water, that's a key driving force for protein folding. Okay, so that's kind of the broad overview of the class. As far as properties of individual amino acids within the class, I think you should know glycine. That's like the simplest amino acid. It doesn't have any R group, it just has a proton there. And the importance of that is that this is the most flexible amino acid, and it imparts the most wiggliness, if you will, or the most degrees of freedom for structures that a protein can adopt. Okay, so the least steric hindrance would be with glycine. That's in contrast with proline. Proline is the only amino acid where you have this bridge between the R carbon and the amine group. So this methyl group, this can rotate around, and the R carbon can rotate around the amide bond everywhere else except proline. So in proline, this bond angle is fixed and you don't have free rotation around that peptide bond. So proline is the amino acid that has the most steric hindrance within a folded protein. Okay, and then let's see, alanine. Alanine, the R group is just simply a methyl. Alanine, independently of its role within a protein structure, alanine has an important role for gluconeogenesis. So just kind of put a pin in that. We're going to have a dedicated lecture on that several weeks from now. Okay, so why don't we take a break and come back at 9:01, and we'll discuss the branched chain amino acids and the other classes of amino acids. Okay.
It's just cool. I mean, I remember a lot.
All I gotta say about that.
I love learning this stuff. Yeah. These are doing so much right
Must be nice piece.
I remember getting those raw.
But then we have
But there's an actual principle.
I have it on my calendar tentatively for Saturday. Yeah, right. But I need to remind which
That was high.
That's a fun
That's so good. I can't have, I mean, I do have dairy. I should have.
Little late, this is the turning solutions session number.
So we just have.
Pass along to your neighbor.
Do you buy the ones physically from Skildum?
Afternoon is 8.
Where is
No, I don't think so. I think I just found one that was like.
Is that also asynchronous? No, I totally. Can you show me the code one more time?
that I just like made a copy of it so people will always see me creep on like this spreadsheet.
Stayed here till like 7, but that was only because when I went home the day before, my Wi-Fi was like down, so I got like nothing done.
I feel like I need to start like planning to stay here later because when I go home, I'm like not
Yeah.
I'm always like, I don't know, take a little break and then I start studying again.
Yeah, you're tired. So if you like sit on the couch or something,
I was just a small gay, bro. Making amino acids.
When
What, how much?
Brandon and Jaja.
our bio.
Those things are just
A little bit, not too bad.
she can just chill.